Тем самым закончим тему, а потом пойдем на обсуждение, чтобы мы поняли, чтобы мы запомнили, да? Давайте. Кто презентует твою презентацию?
Вам видно?
Нам не видно еще. Вот сейчас появилась. Ага. Курманова Габриэла.
Да.
Все, можно.
So I'm going to talk about G protein receptors. G protein coupled receptors, also called as GPCRS, are the largest class of drug targets, largely owing to their druggability, diversity, and physiological efficiency. And many drugs selectively target specific subtypes of G protein coupled receptors, but high specificity for individual G protein receptors may not be desirable in complex multifactorial disease states in which multiple receptors may be involved. And these receptors bind to a wide variety of molecules, including ions, amino acids, peptides, lipids, and nucleotides. They control the activity of enzymes, ion channels, and vesicular transport, principally through the catalysis of GDP to GTP, guanosine diphosphate to guanosine triphosphate exchange on heterotrimeric G proteins. And also they are involved in diverse biological functions, including the senses of smell, taste, and sight, and the regulation of appetite, digestion, blood pressure, reproduction, and inflammation. And it is the reason why they are involved in a wide variety of pathologies and diseases. So here we can see the structure of the G proteins. And structure of G proteins consists of 7 membrane spanning alpha helix structure with an extracellular amino terminus and intracellular carboxyl tail, as we can see in the first scheme. And and represents the largest family of cell surface receptors. They have evolved to detect and transmit a large palette of extracellular signals into virtually every eukaryotic cell, so in human too. And a signal transduction begins when an extracellular ligand agonist binds and switches the receptor from an inactive state to an active state conformation. And activated receptors catalyze the exchange of GDP to GTP on the alpha subunit of heterotrimeric G proteins, which in turn engage conformational events between their subunits. Also, the schematic on the right shows the view from the extracellular side. The helix are arranged in an approximately circular fashion within the membrane plane, and with many receptors, the crevice in the middle of the helix contains the ligand binding site. We can see it here, and which is accessible from the extracellular side, while the extracellular N-terminal domain is short in most G proteins. G protein molecules. Some receptors have larger N-terminal domains, but that may contain ligand binding sites or other functional features. While the extracellular N-terminal domain is short in most G protein receptors molecules, some receptors have larger N-terminal domains that may contain ligand binding site or other functional features. Also, there are two principal signal transduction pathways involving the G protein coupled receptors. Here they are the CAMP signal pathway, which means cyclic adenosine monophosphate. And second one, the phosphatidylinosytol signal pathway. And here we can see two schemes. First relate to the cyclic adenosine monophosphate. And this signal transduction contains 5 main characters. As we can see, it is stimulative regulative G protein, and adenyl cyclase, protein kinase A, and CMP phosphodiesterase. And stimulative hormone receptor is a receptor that combined with stimulative signal molecules, while inhibitory hormone receptor is a receptor that combined with inhibitory signal molecules. Also, adenylyl cyclase is a glycoprotein that catalyze ATP to form this cyclic adenosine monophosphate with the help of cofactors like magnesium ion or magnesium and manganese. The produced cyclic adenosine monophosphate is a second messenger in cellular metabolism and is an allosteric activator of protein kinase A. And protein kinase A is an important enzyme in cell metabolism due to its ability to regulate the cell metabolism by phosphorylating specific committed enzymes in the metabolic pathway. It can also regulate specific gene expression, also cellular secretion and membrane permeability. And if we talk about the second scheme in the phosphatidylinosytol signal pathway, the extracellular signal molecule bind with the G protein receptor on the cell surface and activates phospholipase C, as we can see in the scheme, which is located on the plasma membrane. And the lipase hydrolyzes phosphatidylinosytol into two second messengers. They are inositol triphosphate, and which is also called IP3, and diacylglycerol, which is also called as abbreviation DAG. And IP3 binds with the IP3 receptor in the membrane of the smooth endoplasmatic reticulum and mitochondria to open calcium ion channels. And DAC helps activate protein kinase C, which phosphorylates many other proteins, changing their catalytic activities, leading to cellular responses. So these pathways work on the cellular response, and it's very important when we use specific drugs for these G protein receptors. Next one, drugs that act on G protein coupled receptors. Here I put the table which contains some examples. For example, here I found the dosing of atropine. For the method of application and dosage regimen depends on its form of release and other factors, and the optimal dosage regimen is determined, of course, by the doctor. It is necessary to absorb the complaints of the used dosage form of a strictly specific drug with the indication for use and dosage regimen. And for example, atropine is used to relieve acute pain in gastric ulcer and duodenal ulcer and pancreatitis and in renal. And for example, for children, recommended administration at a dose of 0.01 milligram per kilogram. And for adults, it is 0.25 or to 1 milligram. And there are differences between the age and about the dosing, like in a one time in a day or 2 times in a day. I didn't find information about that because it is very specific due to the disease, due to the individual features of the person. And as we can see here, drug action, they most of them are inhibitors, also agonist and partial agonist. It is due to their like target, their function, during the disease. That's all. Here are the references.
So you took web notes, pharmacology, GPCRs, and CVI, articles, dialogue,
Yes.
so book data, PDF, academic, OCOM. Okay, thank you so much.
Mhm.
Let me stop the recording. Now we can discuss within ourselves.